Okay, well, I am Mariah Valesky. I am a first year genetic counseling student at the University of Minnesota, so I'm up in Minneapolis right now. I did my undergraduate degree at the University of South Dakota, so I am a Yotes fan. Um, but yeah, my undergrad was in biology, specialized in cellular and molecular physiology. And then I'll graduate from here next May with a degree in genetic counseling, which is a master's. So it's a 2-year program.
And don't worry, we talk about her choice of schools often, probably once a month.
Yes, I'd say once a month it gets brought up for sure.
Okay, perfect.
Even though we're in
I
Vermillion
just had to
over
make
time.
that disclosure clear. So,
But yeah, no, it was a good school. Really liked it. Highly recommend, guys. I heard they're getting a nurse anesthetist program that might be applicable to some of you someday. But yeah, so genetics is something I kind of found, I would say, later in my undergraduate career. Originally, I was planning on going to PA school. Very much figured out that I did not love anatomy enough to go to PA school. And so I kind of found my way here. But yeah.
all right.
So if there's no questions before we actually start, I might get started here.
Yep, go ahead. We're good.
Perfect. Okay, so here is like the outline learning objectives that I kind of put together for you guys. So hopefully it kind of flows nicely. So I was going to start just by talking about like the very basics of DNA and how that gets to protein, because I do think that can be really applicable when you're trying to explain that to patients, just to kind of have that core background. So hopefully you've all heard of the central dogma. Basically, DNA getting transcribed into RNA and then getting translated into a protein. So your first step in that is transcription. So that's basically taking your DNA into your messenger RNA. And there are a ton of different RNAs. Messenger RNA is kind of the big one for DNA replication, but just know there are other types of RNAs out there. So that takes place in the nucleus. So if you remember, DNA is double-stranded, but RNA is single-stranded. So the process of transcription is kind of taking that double strand and almost pulling it apart into single strands of mRNA. That way you can copy those nucleotide bases, and then you essentially end up with 2 copies from the 1 copy that you started with. So it occurs in the nucleus. If you have ever heard of DNA, how we read it, we usually read it 5' to 3'. So that's just a way so that everyone's reading DNA in the same order. Because if someone's reading it backwards, that's going to look like a completely different DNA strand. So a little bit more detail than you maybe like really need to know, but just know that for DNA to get transcribed into RNA, it has to become mature. And that involves adding a poly A tail, which is just a bunch of adenosines lined up on the 3' end, and then something called the 5' cap. One thing that is kind of important to realize is that DNA is made up of something called exons and introns. And in order for our DNA to get to its finalized protein structure, all those introns need to be spliced out. So if you can kind of think of it that you have a bunch of exons, or you have an exon, a bunch of intronic sequence, and then another exon, bunch of intronic sequence, and you're pretty much just cutting out all of that intronic sequence. So at the end of it, all you have is your exons, and that's what we call like our coding DNA. So actually, a lot of DNA is intronic sequence. We have absolutely no idea what it does. We know that the mutations in it can kind of affect splicing, but we really don't know what it does. But exons are what our coding sequence is, so that's what we're going to kind of focus on today. So you've added your 3' poly A tail, added your 5' cap, and your introns are now gone. So now it's ready to be transported out of the nucleus and into the cytoplasm of the cell, and it will go find a ribosome because that's where translation takes place. So translation basically reads the nucleotides along that strand, and it will basically insert its complementary base pair. So I'm sure all of you have heard that A's and T's go together, G's and C's go together. So you basically have a polymerase running across your DNA strand, and it just keeps inserting your base pairs accordingly. One thing that I really like to point out that I think is super interesting is that the redundancy of your amino acids kind of leads to resiliency of our genome. So that basically means if you have your 3 base pairs that code for 1 amino acid, there is quite a few combinations of those 3 base pairs that will code for the same amino acid. So if you think about it, there's only 20 amino acids, right? There's 20 amino acids. There's how many different combinations of those 3 base pairs? There has to be some duplication, or there's some amino acids that 3 or even 4 trinucleotides can code for that same amino acid. And that redundancy is what allows for mutations to occur without our genome getting super mutated and disrupted. So I'm sorry, oops, there we go. So this is kind of a picture just to like illustrate that a little bit. So you can see your double-stranded DNA that gets transcribed into that single strand of RNA. And then from there, you're going to your protein. So DNA transcribed to RNA, translated into a protein. So these little, can you guys see my pointer?
Yep, we can see it.
Yeah, oh, perfect. So these little met leucine, serine. Oh, you can't see it. Oh no. I'm flipping all over the place here. Okay. So if you can see down here, the methionine, leucine, serine, those are just amino acids abbreviations. So just remember that 3 nucleotides, so it could be AUG, which remember RNA has uracil instead of thymine. So U and T are kind of interchangeable here. So your very first nucleotides, 3 nucleotides would be AUG, and that codes for methionine. So that very first amino acid. So 3 nucleotides makes up 1 amino acid, and you get a bunch of the amino acids in a row there, and that's what's going to fold into your protein. Does that kind of make sense? Pretty good. I can kind of see head nodding, so I'm going to assume we're good. Okay. Very basics. So here's a little bit better. I'm sorry, I flipped it around on your guys' notes. I swear this is the only change I made this morning. But I think this kind of illustrates it a little bit better. You can see your adenine and your thymine here, and they're bound together by 2 hydrogen bonds. Cytosine and guanine binded together with 3 hydrogen bonds. So why do we care how many hydrogen bonds are holding together our amino acids, or not our nucleotides? It's important because A and T only have 2, G and C have 3. So one of them is going to behave a little bit differently at higher temperature, right? So if it makes sense, it takes a lot more energy to break 3 hydrogen bonds versus 2. So the reason that's important is because when we get into our genetic testing that I'll talk about later, areas that are GC rich, so if you have a lot of G's and a lot of C's lined up together, that's a lot more hydrogen bonds that need to get broken in order for our PCR testing to work. So just kind of something to keep in mind of, like there are areas in our genome that are very hard for us to test, and areas that are very high in GC content are kind of one of those limitations. It doesn't mean we can't do it, but it just requires a lot more kind of finagling with the people in the lab. So one other thing I wanted to point out that I think is super cool. So we only have 4 nucleotides, right? And there's only 4 nucleotides in every living thing there is. But those 4 nucleotides are able to account for all of the diversity in life. But if you think about it, let's say you have a strand that's only 10 base pairs or 10 base pairs long, 4 nucleotides. How many different combinations do you guys think that could be for that sequence? Take a guess. No ideas?
No. Well, a million. I was thinking 4, but I know that's wrong.
Yeah, it is. It's well over 1 million. So 4 base pairs in just a little strand of 10 is well over 1 million different combinations. And that's a 10 base pair sequence. There are trillions of base pairs in your guys' genomes, well, in all of our genomes. So I think that's super cool. But there's kind of like your basics review of DNA to RNA to protein. Okay. So I threw this in last night. I think it's helpful, just so that way if I start kind of talking about things that maybe you guys haven't heard of before, this is something that you can go back and reference. So just to remind you, humans have 46 chromosomes or 23 pairs. Chromosome number 1 through 22 are called autosomes. So you have 22 pairs of autosomes. And the reason I say pairs is because one homologous chromosome came from mom, one homologous chromosome came from dad, and together those two make up one pair, okay? So you have 23 pairs of chromosomes, and we're able to tell all 23 of those apart based off of their banding patterns, but they're not identical. And I really emphasize that because that's kind of what gives you your genetic diversity. That's part of what makes you look different from the person sitting next to you. You all have the same 1 through 23 pairs of chromosomes, but they're different because you got one from mom, one from dad, and everybody has different genes with different alleles on those chromosomes. Okay. So chromosome number 23 obviously is your sex chromosome. So females are XX, males are XY. Alleles are basically alternative forms of genes. So every single person, let's just use chromosome 1, for example. Chromosome 1 has the same genes in the same order all the way down the chromosome. And you have 2 copies of chromosome 1, but those 2 copies of chromosome 1 aren't identical. They're similar, okay? So they have the same genes, but those genes can have different alleles on them. Remember, you got one from mom and one from dad. Odds are mom and dad aren't genetically related, so they're going to have different alleles in those genes. Does that kind of make sense? So every person's gene to code for eye color is going to be on the same chromosome, but some people get brown eyes, some people get blue eyes. So the alleles within those genes can be different, and that's what accounts for your genetic diversity. One other word I threw in here that you guys might not have been familiar with, but you might hear a lot, especially if you do end up working in a setting that's a little bit more genetics focused, but it's de novo. So de novo is a new mutation in the pedigree. So that means, let's say you have a child with Duchenne muscular dystrophy. It's a mutation on the X chromosome because it's an X-linked condition, so it's a lot more common in males. But mom didn't have that mutation. So we went back, tested mom. She doesn't have the mutation. Her son does. So that's what you would call a de novo mutation. It arose from something that didn't exist in the previous generation. A germline mutation is a mutation that's passed down from the gametes. So that's kind of almost the opposite of a de novo mutation. It's something that came from your parents. It was some mutation that was in the sperm or the egg that was passed down from parent to offspring, which is different from a somatic mutation. And that's something that occurs in any cell that is not your gamete. So the big example for somatic mutations would be like cancer. It's a mutation that arose well after the child or person or whoever is born. And it was a mutation that happened that caused like a cascade of events to make those cells kind of divide like however they want without any checkpoints. And we'll talk about that more later, but just know that since it's a somatic mutation and it's not in the gametes, that mutation cannot be passed on to the next generation. And then the last one that I wanted to point out to you guys is a novel variant. Similar to de novo, and that's the right reason I put it on here is because that's kind of a term that maybe gets interchanged a little bit too loosely. They're two different things. A novel variant is a new mutation that's never before been documented, never before been seen. It might sound very rare, but honestly, it is pretty common, especially with what I see all the time. So novel variant, never seen before. Those can be tricky because you can kind of guess what that variant might do based on where it's at, but really it's never been studied before, so we don't know. Okay, so chromosomes. So we talked about DNA to RNA to protein, and now these proteins can get coiled up into chromosomes. Okay, so these chromosomes are just super densely packed I want to say pieces because they are kind of like pieces of DNA. That's not like the best term, but like bacteria have circular DNA, so they don't have chromosomes, right? All of our DNA is linear. So it is almost like pieces. Like if you stuck them chromosome 1 up to chromosome 2 up to chromosome 3, it would form a linear line. And that's kind of what makes us different from bacterial DNA, because theirs is circular. There's no way that theirs would be condensed into a chromosome-like structure. Something I do want to point out, kind of throwing you guys all the way back to the cell cycle, you can only see chromosomes when they're going through either mitosis or meiosis. So if you remember during those two cell cycle replications, the DNA has to be tightly coiled, so that way when it gets pulled apart into two new cells after it's replicated, you end up with the right number of the right chromosomes in different cells. So you can only see them in dividing chromosome or in dividing cells, and that's important because when we go to test for different chromosomes, it takes a long time because we have to culture those cells and wait for them to be dividing. So something called karyotyping is basically a snapshot of the chromosomes. So that's how we look for trisomies. So trisomy 18 and 13, not as common. I'm assuming most of you have heard of trisomy 21, which is Down syndrome. So that's like one of the most common non-disjunction events that we'll test for. But we basically take a snapshot of all the chromosomes and we can look for an extra copy of chromosome 21. But that test takes a while because we have to wait for cells to divide. So it has to be cultured. Other things we can look for, like sex chromosome aneuploidies. So Turner syndrome, which is that single X, Klinefelter, which is that next XXY karyotype, or triple X or XYY. That's kind of a lot, but just know that when we do karyotyping, everybody is expected to be euploid. So everyone is expected to have 46 chromosomes. And when we find something that's more than 20 or more than 46 or less than 46, that's something called aneuploid. So it's just not what we expect for the normal number of chromosomes. Triploidy is having an extra copy of every single chromosome. So you'd have 3 copies of chromosome 1, 3 copies of 2, 3 copies of 3.
Is that pretty rare?
Incredibly rare. It is probably life limiting. I think the oldest I've ever heard of someone living with it is maybe a year. Most of them would miscarry before birth, but it is possible. The example that I was always told about that is think about baking a cake, okay? So the reason why triploidy can happen versus some other aneuploids, so let's say you're baking a cake and you put in too much flour. Your cake's going to turn out really, probably not great, right? Like something went wrong, you added too much of something that was important, and now your cake didn't turn out right. Well, with triploidy, think about baking a cake, but you accidentally double the recipe. The ratios are all still right. You just have more than you would expect, right? So that's the reason that triploidy can happen versus some like trisomy 22. That one would never be viable because chromosome 22 is such a gene-rich chromosome. It just wouldn't survive up to birth, I would say. Okay, so here's the karyotype I was talking about to you guys. I think it's a lot easier to see. So this is that picture on the left there is what it would basically look like when a cytogeneticist was looking at all of the stained chromosomes. And then people much smarter than me are able to look at all of those chromosomes and sort them into the picture on the right. So they do that off of their banding patterns. It's called G banding. I think it's like Gizmo banding. We call it G banding. I think I've only heard that word once in my life. So G banding. You can kind of think about it like a barcode. Each chromosome should be stained in a very certain way based off of how gene dense or gene light it is. I cannot do it. I could maybe pick out like one of them, and that would be solely off of size. So people that are trained very well are able to do this very well. I do think they have computer softwares now that can sort them, and then they have cytogeneticists go in and like double check the computer, but that's how it's done. But that's how we can look for aneuploidies, sex chromosome aneuploidies, triploidy, anything like that. So take like 10 seconds and look at this karyotype and tell me what condition you guys think they have. Where is there something that there shouldn't be?
Is that the general consensus? You guys see that? Okay. I think they all said 21, so.
Yep. So this is a trisomy 21 karyotype, which would be diagnostic for Down syndrome. And then what biological sex is this patient?
Girl.
Yep, it's a girl. So you can see there under the sex chromosomes, those are 2 X's. If it was a male, it would have one of those, and then the Y is very small. I'd say it's about the size of chromosome 21. So the Y is just itty bitty. You can see these are 2 full-size X chromosomes.
So that little curve on the first chromosome really doesn't mean anything because it's very similar to the one to the right of it, correct?
Yep, they would only look at the banding pattern, not so much like the curvature or anything. Yeah. So I mentioned earlier that we wouldn't be able to have trisomy 22, which is kind of surprising, right? It's like one of the smallest ones on there. It's not that important, right? But if you look at it, look how dark it is on the Q arm. So the P arm is the short arm, stands for petite. The Q arm is the long arm. So the bottom half of that chromosome, look how dark that is. It's super, super gene dense. And that has huge implications for trisomies because it's just not compatible with life. Chromosome 1 is the biggest, if anyone wasn't able to pick up on that, but size doesn't always mean it's going to have more genetic material or not. Okay. So epigenetics, super, super complex topic. I put it into one slide for you guys, mostly because I think it's something you should be aware of. It's something that absolutely impacts genetics, but I don't think it's anything that you guys need to understand thoroughly. I don't even think like geneticists understand it thoroughly yet. But just know that epigenetics is something that changes genetic expression due to genetic modifiers, not because your DNA code is different, but because you have modifiers on your DNA. So epigenetics is a huge reason why siblings look different and why even twins can look different. So let's say you have a identical twin. You guys they would have the same genetic sequence, but you can still kind of tell them apart, right? There's no identical twins that look so identical apart, not even their parents can tell them apart, right? And that's due to their epigenetics. There is some research saying that epigenetics is heritable from their parents. I mean, I do think it's true, but we just don't know a lot about it yet. So the main one that you guys should know about is DNA methylation. That's basically where a methyl group is attached to the DNA and it causes repression of transcription. So remember, transcription is DNA to RNA, and having that methyl group pretty much stops that. So it silences your gene expression. There is evidence, like I said, it can be passed on via mitosis. Something that you should be aware of is that the methylation should pretty much reset with each generation. So in your gametes, so your parents' gametes, or however you want to think about it, in the gametes, that methylation should pretty much be removed. So that way, when an egg and a sperm are fertilized, the methylation process starts over. And the reason why I mention that is because when that methylation isn't removed in the gametes, that can lead to imprinting disorders. So if any of you have heard of Angelman syndrome or Prader-Willi syndrome, those are both imprinting disorders. And they occur at the exact same place on chromosome 15. But if the demethylation didn't happen in the dad's chromosome, it leads to Angelman. The demethylation didn't happen in mom's chromosome, it leads to Prader-Willi, which I personally think is crazy. It's 2 completely different disorders. They look very different, but same chromosome, same location. It just depends if the demethylation happened with mom or with dad. Histone modification, that's basically just how tightly the DNA is wound. So euchromatin is super loose DNA, heterochromatin is super tightly wound DNA. Heterochromatin, it's so tight that transcription factors can't get in there, so it pretty much represses the gene. The big example for that, if you remember, females have 2 X chromosomes. You don't really use the second X a lot. I would say like 10 to 15% of gene expression from your second X is all that's used. The rest of it is stored in a heterochromatin form. Okay. Chromatin architecture, that's just kind of the catch-all for anything outside of histone modification and methylation. Just know that these aren't the only 2, but they're kind of the main 2. So I just wanted to mention to you guys, if you ever run into Angelman or Prader-Willi, 2 of the still incredibly rare imprinting disorders, but 2 of the more common ones when you're talking about imprinting. All right, so mutations. Hopefully you guys have maybe kind of seen this before. So a frame shift mutation, remember when I was talking about your polymerase is running along your RNA strand to make it go from single-stranded to double-stranded. A frame shift mutation occurs when there's a mutation in your coding sequence that is not a replicate of 3. So remember, 3 nucleotides make up 1 amino acid. Well, what if there's a deletion of one of those nucleotides? Just 1. But now your polymerase is going to go along, and it's all of a sudden when it hits that deletion, it's going to read another set of 3, even though 1's missing, but it still has to read 3. So now from that point all the way down, your reading frame is going to be changed. So now you're going to get amino acids that aren't supposed to be there. You're going to be missing amino acids that you need, and it basically just messes up the rest of that reading frame. And with that, it can also mess up your stop codons. So stop codons are at the very end. It basically tells your polymerase to fall off, that it's done. But if it doesn't read that stop codon because now you have a frame shift mutation, it can continue to go on further and further, which is going to lead to a massive protein when you only needed like a small one. Or it can also lead to a nonsense mutation, which is where it basically accidentally runs into a stop codon too early. Okay, so there's...
I think one explanation, correct me if I'm wrong, but with frame shift, with any mutation, what she's mentioning is think of a zipper, as you're unlocking that DNA. If one of those little teeth are wrong or incorrect, it's going to continue to replicate that all the way down that DNA until that stop codon says, okay, you're done, and then just sends it off to do its work.
Yep. No, that's exactly right.
That makes sense?
And
okay.
Yeah. And the big thing with frame shifts is if you miss that stop codon now, because it's not there, it's going to keep going into your into your next reading frame. And that's going to just have a whole cascade of effects. So frame shifts are usually pretty significant mutations. Nonsense ones is basically, like I kind of mentioned, 1 nucleotide got replaced, and now that amino acid it's reading for, or coding for is now a stop codon. So now your polymerase is reading, and now there's a mutation, and it stops here when it should have gone all the way through. So now a good chunk of your protein is no longer getting translated anymore. So it basically stops too early. Obviously, the severity of that kind of depends on where it happens. If it only stopped a few amino acids short, it might be okay. Maybe those amino acids at the end were really important. But if it happens kind of in the middle of a gene or early on, that protein is going to be pretty small compared to what it's supposed to be. A missense mutation basically is, if you guys have ever heard of a point mutation, same thing. It's basically where you might have 1 wrong nucleotide in there, but it doesn't alter the whole reading frame. It just changes that 1 amino acid. So maybe, actually this picture here is a missense example. So this is actually the mutation for sickle cell disease. So something I did not realize until I got here is that sickle cell disease in everyone is this same missense mutation. If it's not this missense mutation, it's a different type of beta thalassemia. But it's basically where your polymerase is going along. See where it says DNA and it's normal. So it should code for a glutamine, which is the protein at the bottom under or above the normal protein. But your polymerase is going along, and now there's a substitution, and it's now coding for a valine. And that's going to lead to sickle cell. Okay, so it might not seem like a lot. It might just seem like one amino acid, but it can still have huge health implications. A synonymous variant or mutation is a change in the DNA sequence, but it still codes for that same amino acid. So I'll use the same example here. If we are going along and we're supposed to code for a glutamine, but now instead of having, let's just say this switched it out for, I don't know, a C. So we have a C and a G instead of a T and an A. But that still codes for glutamine down here. It's the wrong nucleotide, but you're still getting glutamine. So for the most part, those are fairly benign mutations.
Which are the mutations that you see probably most often?
Like in severity or just most commonly?
Probably most common and then most severe.
I would say missense is probably the most common.
Okay.
Most severe, I would probably say nonsense mutations or these del dupes. So if anyone's heard of Huntington's disease, it's due to a trinucleotide repeat, which basically gets too large, it becomes unstable. It kind of depends. They're also kind of in different classifications. So del dupes are considered structural variants, where nonsense mutations would be considered sequence variants. So they're almost like two different classes of things.
Okay.
But yeah, anything that kind of causes a premature stoppage usually is pretty severe. Okay, so del dupes, like I kind of just said, they're considered structural variants, not that you guys really need to know that, just know that they exist. And it basically means that you're missing a huge chunk of DNA or you have way too much DNA that got duplicated. So I said a good example of a duplication is Huntington's disease. Another one is Fragile X disease, or Fragile X syndrome. That's basically where everyone has these trinucleotide repeats. Let me think here. Fragile X is CCG. So in Fragile X, I'm just going to use that example because I remember the repeat. In Fragile X syndrome, normal would be everyone has less than 100 trinucleotide repeats, let's say. Okay, so it's normal for you to have 80 repeats of CAG in your coding sequence. But when you kind of get past that 100, now that gets to be unstable, and that's what causes Fragile X. So duplications, super common in your genome. Everybody has them. But when they get to be too big, that's when they get to be unstable. It also depends where they're at. Like there's parts of our genome that are meant to have those trinucleotide repeats, and then there's some where a mutation occurred and now you just have a giant duplication, and that's now problematic in terms of disease. The last one I'll talk about is an insertion. It can be the insertion of one base pair, which if it is just one, now you're leading to a frame shift mutation. Let's say you inserted 3, so now you just have an extra amino acid in there. Not the end of the world, probably, but you know, you just never know. And location is such an important part on that. I do get asked, like, what's the difference between a duplication and an insertion? Honestly, it's like kind of the same. Like technically a duplication, you're inserting DNA. It just happens to be a duplication of another piece. So definitely kind of a gray area, but if it's not like a sequence that is already found in our genome, it's not really considered a duplication. So just be aware when things like deletions, insertions don't happen in groups of 3, that's when you kind of lead to a frame shift. So inheritance pattern, These are like the three most common that you'll probably see. Dominant, so dominant only requires one mutation in one of your chromosomes for it to cause disease. So you're going to see that mutation continually be passed down from one generation to the next. So you'll see it in successive generations if it is a true dominant disorder. Males and females should be affected equally. Homozygotes, so if it happens to be an autosomal dominant condition, but now you have mutations in both of your chromosomes, a lot of times that's going to lead to a lot more severe disease, or it might even be lethal before birth or shortly after, really just depending on the disease. And then if you have a parent that is affected with this autosomal dominant disease, that's a 50% chance of passing it down to either a son or a daughter. So you guys can understand how that math comes into play there.
So for an example, if I were to ask you a question about inheritance, and I said a patient is diagnosed with a dominant mutation, a dominant or recessive, what's the correct term I'm looking for? Dominant inheritance?
Yeah, like an autosomal dominant inheritance.
So if I said an autosomal dominant inheritance, and they're talking about to their son or daughter, you can see that they have a 50% risk, okay? Whereas as it goes further on, you can see parents healthy, they have a generally a 25% risk to each son or daughter. So this is what is really important when we're talking about diagnoses of different disorders. What are the risks to the offspring? Okay, so this is going to be important when you're discussing the positive results with your patient. What does this mean for my kids? Okay.
I think, can you guys see that up there? I almost feel like it's easier for me to draw.
Yeah, I can see you're ready now.
Okay, perfect. I'm very much a visual person. I don't do well with words. So if you're like me, hopefully that's a little easier. I don't have a different color. That was our issue. All right. So here is, let's say, mom's chromosome 7. I don't know. Doesn't matter. So mom's chromosome 7. You can see my little highlighter mark there is her autosomal dominant mutation. Okay. Remember, you get one chromosome from mom, one chromosome from dad. So you can see here how dad will pass down a healthy chromosome 7, right? Because he doesn't have a condition. But now mom, you have to get one of those from her. So it's a 50-50 chance whether you're going to get the good chromosome or the one with the mutation. So if you go into your, does that make sense? Everyone's good with that? I think, yeah, I'm going to assume we're good if we're done. If I don't hear anything. So then if you're talking about autosomal recessive conditions, this is kind of more important when you think about parents that are both carriers for the same condition. So I'm going to use cystic fibrosis because that is one of the more common ones that most people are familiar with. So now you have cystic fibrosis chromosome 15, I think. We're going to go with 15. So now on the left there, you have dad's chromosome 15. He's a carrier for cystic fibrosis. Little highlighted mark there, okay? Mom, also a carrier for cystic fibrosis. On the right there, okay? So now there's a 25% chance that they're going to have a son or a daughter with CF, because there's a 50% chance that dad passes his affected allele. Whoa, sorry guys. I can't get rid of it. Oh wait, yeah.
That's okay.
There we go.
You can leave
All
it.
right, 50% chance that dad passes his, 50% chance that mom passes hers. So you add those together. Nope, multiply them together. 25% chance that they're going to have an affected son or daughter. Does that make sense? So if you guys ever saw Punnett squares, that's where this comes from.
Are you guys struggling with that? A little bit. So to make it a little bit more, so the 50% comes from that one parent with the dominant. With recessive, each parent has a 50% chance of passing it on. So if you have 50% chance plus 50%, you get 100%, right? But you have to divide that by 4 because there's that potential of 4. So it's 25%. Helpful? Some people. So basically what you're getting is you're getting a 50% chance from one parent, a 50% chance from another parent. So the overall risk is 25% because it's 50 and 50. So it's not like multiplied by 2, it's divided by 2.
So what about the dominant? Both parents are dominant, will they get it?
If both parents are dominant, that should express completely, correct? That should be 100% if they're both dominant.
Okay, so a good way to look at this is called the Punnett square up here. Given my drawings, not great, guys. I apologize. But same thing, we just moved mom up here. Dad's over here. Okay, so these are both dad. Mom's on the top. Here, there's a 25% chance they're going to have a completely healthy child. Here, there's a 25% chance that they inherit, or the child inherits both mutated chromosomes. And then these other two options are carriers, which CF, not a huge deal. There's a ton of carriers for CF. But do you guys kind of see where that 25% comes in now? I think Punnett squares are maybe easier to look at, but does that kind of help? Yeah. Yep.
Yeah, I understood the 25% and the 50%, but the Punnett squares are catching me off guard, but that's okay.
Oh, I'm sorry. Okay.
No, not your fault
If it
at
doesn't
all.
make sense, ignore it.
Nope, no, not at all. I think I saw a lot of heads nodding after you listed that, but I just remember percentages, so that's how I remember. So go ahead. I'm sorry.
Yeah. Okay, so we're gonna move on to X-link now. A little more complex. Not much. It's about the same. But okay, X-link. So remember, mom has XX chromosome, right? Dad has XY. Okay. So X-linked, you need to have. So this is technically, if you think about it, X-linked recessive. Okay. There's not really an X-linked dominant that I can think of. So that's why I only put, when we talk about X-linked, we're almost always thinking X-linked recessive. So remember, recessive, I said that you have to have 2 affected chromosomes to manifest the disease. Well, females only have 1 X chromosome, right? Or excuse me, males only have 1 X chromosome, right? Their other one's a Y. So if their 1 X is affected, that's all it takes for them to manifest disease. So the example I used earlier is Duchenne muscular dystrophy. It's a really severe degenerative neuromuscular disorder. I would say most people pass away in their early 30s, maybe 40 if they're lucky. So it's a very, very severe disease, but it almost only affects males because the mutation occurs on the X chromosome. So it only takes 1 mutation for a male to manifest that. Where a female, so if they inherited it from their mom, the mom would only have that 1 mutation that got passed on to their child. Okay, so that's why if you look at a pedigree, which is basically the family tree, in an X-linked condition, you're going to see almost all of the males are affected. You're not really going to see any affected females. That's because females can be carriers for these conditions. It's possible that they have a mutation on both X's, but it's incredibly, incredibly rare. But it's not uncommon for a male to develop that disease because all it takes is for them to inherit that 1 mutation that their mom carried.
So if I were to ask you a question about an X-linked condition, and I said that the parent's son was diagnosed with Duchenne muscular dystrophy, which is an X-linked issue. Now, the patient asks, or the mother asks, will my daughter contract this? What would be your answer? No, because it's an X-linked, so it only affects males. So that would be an example of a question that you would talk to a patient about. Will all of my kids suffer from this condition? No, the only ones that are at risk would be males. Okay, that clear it up a little bit, make it a little bit more sense. Okay, right, they can still be carriers, but they will not express that. Okay.
Are men who have the disease sterile? Because if a male that has it, a female that's a carrier that has a kid, then the kid's more than likely got a 25% chance of having it.
Yeah, I don't know about sterility, but I do know with like Klinefelter syndrome is when you have that XXY chromosome. That's when you start developing sterility, and you have male expressed sex characteristics, but they're generally infertile.
Yep.
I
With
think
DMD, there isn't really
I think everyone needs a break.
okay.
I think
That
everyone
sounds
needs a break. Let's take 10 minutes. So I'm going to shut my camera off. We'll come back at 11. So
That's good. Okay, can you guys see my whiteboard?
okay.
Whiteboard? You can see this right here.
Yep, we can see that.
Okay, perfect. I think this will help you guys. I will say I'm more of a visual learner, so maybe if percentages make more sense to you, then this might not be quite as helpful. But are we good on recessive? Do you just want me to do X-linked?
I think
Or do you
so,
want me to
since you did recessive already, but if you could do everybody a favor and kind of post this information or send it to me so that I can show kind of what this looks like.
Yeah, absolutely. I can make you a lot nicer one than this too. But for time purposes here.
Sure.
Okay, so we're going to look at the X-linked one here, okay? So for disease to manifest, females need to have 2 mutations, okay? Males only need 1 mutation for disease to occur. That's because males only have 1 X chromosome, okay? Females need 2 because they have 2 X's, okay? But males only need 1. So you can see here with my blue XY, hopefully this makes it a little easier if it's color coded. Blue XY, male, female, pink XX, but this female has a little red mutation on one of her X's. See that? Okay, so she's a carrier for the disease. We'll say Duchenne's because that's what we've been talking about. She's a carrier for Duchenne's. She doesn't have Duchenne's because she would have needed to have a mutation on her other X, but she doesn't. But remember, there's a 50% chance that she's going to pass down her mutation. All right, I lost my ability to write.
As long as you send that information to me, we can move on. So
Okay,
yeah,
sounds good.
I can post that. So
I'll make you guys a nice one to follow. Hopefully that's easier. Can you guys hear me?
yep,
Yeah.
we can hear you.
Okay. My laptop is frozen. Love that. Give me 2 seconds.
All right, I'm gonna stop your screen sharing just so when you're ready, you can start with your presentation again.
Okay, sounds good.
And we'll take another break. Yeah, it's like 2 years ago. A year and a half. There's a
Okay, did we lose you?
I can hear you guys fine. I can't do anything on my screen though.
Okay.
All morning it worked just fine. Let me.
You want to just log off and then pop back on? Maybe that'll help.
Yeah, that's what I'm trying to do here.
Okay.
I might need to.
after you said. Yeah.
So weird. It shows that you're still here. I don't know what's going on. Okay.
Okay, I think we're back.
Okay.
Sorry, guys. All right, we're back. I've never had that happen before.
Do you want to just move into the prenatal genetics piece of it?
Yep.
Okay. Actually, Mariah, when I'm thinking about it, maybe let's just move to the cancer genetics, because they're going to talk about newborn stuff next week. So maybe to keep things going, we just need to finish up by like 11:30.
Okay, perfect.
Okay. Can you still share?
I don't know. It must be something with the, sir, I might have you screen share and I might just present off of you if that's okay.
That's perfect. You just tell me when. I'm going to start at the cancer genetics, and then we'll go from there.
Perfect.
Okay. Can you see me?
No, my audio works. I can't see anything.
Okay. All right.
There we go. Okay,
Okay.
we're good. Holy cow. Sorry, guys. Okay. All right, so we'll go into cancer since you guys are doing prenatal next week. But you guys have access to my slides too if you are curious about prenatal screening. They're pretty detailed, so hopefully you can read through it. So I threw this in here. This is from the American Cancer Society. It's from 2011. I like this table better because it's easier to look at it and it's a little bit more condensed. But as of 2021, the stats haven't changed. In their lifetime, 1 in 2 men will develop cancer and women 1 in 3. So the summary from this whole slide here is that cancer is super, super common. Go ahead, sir. So I also just put this in here so you guys can kind of see what the most common was. Prostate cancer is the most common for males, while breast cancer is the most common for females. And then if you look down the line, it's honestly pretty similar. The melanoma of the skin and the kidney, renal, pelvis cancer is actually the next 2 for females. So other than prostate, breast cancer, fairly similar cancer forms in both males and females. Go ahead. So the one thing I wanted to talk to you guys about is cancer susceptibility genes, because I do think that'll be super applicable to you guys in your nursing careers. So basically our cells have 3 big checkpoints, okay? Oncogenes, which basically regulate proto-oncogenes, and those are just super big words for proto-oncogenes basically promote cell growth. Oncogenes regulate that. So when there's a mutation in an oncogene, that regulation of those cell growth genes are no longer happening. And that's kind of what causes that uncontrollable cell growth that you see in cancer. So these are typically gain of function mutations. You guys will hear that a lot in genetics, gain of function versus loss of function. So here it's a gain of function because these cells are growing uncontrollably. There's nothing to like prevent that or to stop it or regulate it anymore because now those oncogenes are not functioning. So a big or example of this is multiple endocrine neoplasia type 2, which is just a type of cancer. But that's due to a mutation in an oncogene. The second one is tumor suppressor genes. So these are genes that pretty much make proteins that suppress tumor growth. So very similar to oncogenes, just kind of a different type. The biggest difference between these and oncogenes is that these are responsible for loss of function mutations, So the big example of this is your BRCA1 and BRCA2, which is your primary gene responsible in breast and ovarian cancer, also a huge impactor in prostate cancer for men. So these genes help regulate cell growth, so that way tumors aren't forming throughout your body. But when you knock out this checkpoint, that makes you that much more susceptible to developing that. And then your third one is DNA repair genes. So DNA repair genes make proteins that help your cells repair DNA damage. Damage occurs throughout your lifetime, whether that be from some type of teratogenic exposure. It happens from being out in the sun, all those types of things that cause DNA damage. These proteins are here to help repair that. But when you have a mutation in one of these DNA repair genes, your body is no longer able to repair that damage, and that makes you that more susceptible to cancer. So those are kind of the big three that play into hereditary cancer development. If you want to go to the next one, I think that summarizes it. So this is kind of what I wanted to get at with you guys and how this will be super applicable to you working with patients. So whenever we see patients, the big question we always get is like, why was I the one that got cancer? Why was I the one that developed this? I have how many siblings? Like, they're all healthy, or kind of things like that. And so this is kind of like a super simplified diagram that I think kind of helps illustrate that. So if you remember that very first table we looked at, your cancer risk increased as you got older. And that makes sense, right? Because your body has had that much more time to acquire damage, for genes to get damaged and not able to be repaired and things like that. And that's why you usually see cancer that's not hereditary develop a lot later in life. Well, if you were born with a mutation in an oncogene or in a DNA repair gene or a tumor suppressor gene, those three we just talked about, now you're one step closer to already developing that cancer cell. And so it's basically like you knocked out one of those checkpoints, and now you only need to get, you only need one more thing to happen before you're starting to acquire cancer. And so that's kind of the explanation we try to give to patients. There's something called a two-hit hypothesis that basically means that two bad things have to happen before cancer starts to develop. Well, if you inherited a mutation because your family had a BRCA mutation, now you only need one mutation, or one sporadic something to happen before you start developing that cancer. So that's kind of the explanation we like to give to patients. When we're in clinic with them, we a lot of times will like draw this out step by step and cross one off and say, everybody started here, but you ended up starting here because of your hereditary mutation that you got. It's not a guarantee that cancer is going to develop because they have this mutation, but it's just a lot higher chance. And then because of that, we tend to see hereditary cancer start at a lot earlier age, which makes sense, right? Because you need less time for that mutation to manifest into cancer. So how much of cancer is hereditary? The answer is not a lot. You can see that 70 to 80% of cancer is just sporadic. So it happened in those somatic cells that we talked about at the very beginning. Hereditary cancer only makes up about 5 to 10%. So the BRCA1 and 2 is honestly a giant chunk of that. Lynch syndrome is pretty rare, but that's a very severe cancer, hereditary cancer. If you guys ever see that, it causes about any kind of cancer that you can imagine. So it is pretty severe. And then familial clusters are kind of something that I wanted to mention too. This is where you can see clustering of cancer in a family, especially in siblings. Maybe the parents are unaffected, grandparents unaffected, but now you have like 3 cancers in a group of 4 siblings. That makes up about 15 to 20% of cancer. And that's where we cannot find a genetic mutation. We don't know why this group of siblings is more susceptible to developing cancer, but for some reason it happens. We can't find a genetic cause, but also a big thing that research are starting to look at now is environment. So we believe that the familial clusters are kind of a combination of genetics, and then a lot of times siblings are sharing the same environment. So that plays a big piece into it. It's just that we can't find a genetic cause yet. So if you guys are going to take anything away is that we are fairly good at testing for hereditary breast and ovarian cancer and colorectal cancer. And that's about it, which is honestly, I think, a little bit scary. We're getting better at testing for other things, and there's definitely susceptibility genes that we know of for other types of cancers, but these are the ones that we can effectively test for fairly well. And we have good guidelines as to how we can change medical management. So red flags for hereditary breast and ovarian cancer that you guys should take away from this is any type of breast cancer that started before age 50. So remember I said most hereditary cancers start earlier. That's the same case for this. So breast cancer before age 50, ovarian cancer at any age, just because ovarian cancer is so much more rare and it's usually associated with a BRCA1 mutation. A male breast cancer at any age is always a red flag for us because it's a lot more common in females because estrogen promotes the growth of cancer in the breast cells. Males obviously have a lot less estrogen than females do. So if there's a male breast cancer, that's usually indicative of a mutation. If you have multiple primary cancers, or the patient is Ashkenazi Jewish ancestry, the reason that's a big one is because that's what we call a founder population. And founder populations are typically ethnicities or ancestries that were very isolated. So they typically tended to reproduce within that population only. And so people with that ethnicity tend to have a lot higher mutation rates. So in the Ashkenazi Jewish population, there are 3 BRCA mutations that 1 out of every 40 people in that ancestral group will carry a mutation for. So that's why we kind of like pull out that ancestral group because they're known to be mutation carriers within that. And that's the case for a lot of hereditary mutations. If we know there's a founder population, a lot of times people can qualify for genetic testing solely based off of ancestry. And then if you have a relative, obviously, that's known to have a mutation. And then, like I said, colorectal cancer is kind of the other one that we can effectively screen or do genetic testing for, and we have solid cancer guidelines for. But these are kind of the red flags for that in a family history. If you see cancers in 2 or more close relatives, if there was a really early age of diagnosis, so that tends to be under 50, multiple primary tumors, or kind of like we call it like a constellation of tumors, that's a lot of times indicative of a specific hereditary syndrome. And then if there's evidence of autosomal dominant transmission. So remember, we said in autosomal dominant that we kind of tend to see that in every single generation. That's usually kind of a big red flag for colorectal cancer because there is only one autosomal recessive condition for colorectal cancer inheritance. So when should people consider genetic testing for cancer? If the patient has a reasonable likelihood of carrying a cancer mutation, we kind of tend to say the rule of thumb with that is there's a higher chance of finding a helpful mutation versus a mutation that we don't really understand. And I'm going to talk a little bit more about that in a couple slides here. If there's a genetic test available that can adequately be interpreted, again, I'll talk about that more in a second, but there's a lot of things out there. Yeah, here we go. So that third one, sorry, that third column there is a VUS. And so a VUS is basically a variant that we know isn't supposed to be there, but we don't really know its impact. And so if we're doing genetic testing and we're just coming up with VUSs, that's not really helpful for anyone because we can't make medical decisions off of them and we don't really know what they mean. So that's what we mean by adequately interpreted. We have to be able to interpret the results that we're getting. If those results will influence medical management. So a lot of times, especially with breast and ovarian cancer, we're talking about like prophylactic surgeries. So double mastectomies or total hysterectomies, things like that. And then a lot of times the patients just want that information. If they've had a family history of cancer and nobody really knows why, a lot of times that can give them a sense of empowerment, finally being able to put a label with whatever cancer that's been running in their family. So genetic testing results. So if we test for a mutation, these are like the 3 possibilities that we can find. So the first one's obviously a positive result. So we found we were looking for a mutation and we found it. For the patient, that finally means that they have an answer. For a lot of times, this can be years of ongoing testing for patients. Our genetic testing has significantly gotten better in the last 5 years, I would say. So this patient finally has an answer, and a lot of times that's been a long-awaited answer for them. But now what does that mean for the family? Is there a risk to siblings? Is there risk to children? How does that kind of look in the family dynamic? How can this result now influence medical management? So like I talked about earlier, prophylactic surgeries can be a big part. And then how will knowing this information impact the patient? So I will say this tends to be slightly less of a cause in cancer genetics, just because most of the patients we're seeing already have cancer. I would say in terms of like neurological disorders like Huntington's, if we tend to find a mutation like that, it's usually like maybe when the patient first started developing signs of ataxia and things like that. So now they're going to have to go the rest of their life knowing that they're going to develop this degenerative disorder. A negative result, I have a little bit more about this in kind of like my last slides for you guys, but is it a true negative or is it a negative because we couldn't find the mutation? And I would say this is one of our biggest limitations in genetics right now. We have to know, like the patient you're working with isn't always the best person to test first. Because if there's a hereditary cancer in the family and your patient doesn't have cancer yet, do they not have the mutation, or were we just not able to find the mutation? So that's where testing family members can be a really important part of genetic testing. A negative result doesn't guarantee the future health of the patient or the child. So again, I'm going back to cancer, but let's say they have a family history of a BRCA1 mutation and we tested affected relatives, our patient came back negative. We're fairly confident saying that this is a true negative result. But remember, 1 in 3 women, just off of population risk alone, are still going to develop cancer. So just because you don't have this one mutation doesn't mean that you can't go on to develop cancer later in your life, just out of sheer like not being lucky, just because cancer is so common. And then follow up. So if it's a negative because we didn't know what to look for, we're constantly finding new genes that are implicated in cancers or cardio or neuro or you name it. This field is rapidly expanding. And so we do encourage patients that we weren't able to find a mutation to follow up, because in a couple of years, maybe there is like a new panel we can run for them. And then the third one I talked about is a VUS, so a variant of uncertain significance. So we were looking for something, we found this, we don't know what it is. Can that change? Absolutely. So these can be really difficult. They can be hard for patients to deal with. They can be hard for genetic counselors to try to explain to patients. But this is why your pre-counseling before testing is such an important thing, because patients need to be aware there's always this chance of finding it. There's definitely a chance that in the future, this variant could be reclassified to pathogenic. There's also a chance it can be reclassified to benign. You just never really know. And it's kind of something that patients just have to accept as a risk with genetic testing. But that's why it is important to do a proper like informed consent. And like I said, medical decisions should never be made on a VUS. I will say there are probably exceptions to that rule somewhere, and I've definitely heard about them, but that tends to be more in like cancer and cardiogenetics than like a prenatal setting. So here's kind of your ethical piece for the day. I'm sure you guys have heard all about ethics and genetic testing, but a lot of times, especially with autosomal recessive conditions, we need parental samples. And that's to kind of determine the phase of the variant. So if we see 2 variants in our patient sample, did 2 of them come from mom and none from dad, or did 1 come from each? So just so you're aware, the lab will always run a paternity test on the samples. Even if they're not looking, they will very easily find out if dad is not really dad, just because the genetic material would be so different. So that's something that needs to be disclosed in informed consent, because if there is non-paternity, it will absolutely be discovered. Secondary or incidental findings. So ACMG, which is the American College of Medical Genetics, so that's kind of who we fall under, has a mandatory reporting list of about 48 genes. Those are genes that if they're found on a genetic test, even though we weren't looking for them, we just happened to find them. That's ACMG used to mandate that we put it on the patient report, regardless of what the patient wanted. Now, I want to say it got changed a few years ago, but now patients can opt out of incidental findings. I will say all the genes on that list are actionable. So they are all genes that have the ability to screen or increase screening or treat or something like that. It's not like the gene for Huntington's where we have no cure. We're not even looking for that unless that's what the patient is there for. These are like BRCA1, where we have good screenings for breast mammograms or breast MRIs, where we can actionably increase medical management based off of these findings. But that's also why a pre-test counseling is so important, because the patient needs to be aware that unless they tell us otherwise, if we happen to find one of these things, we're going to put it on the report. GINA, which is the Genetic Information Non-Discrimination Act, basically protects people for medical insurance, regardless of genetic testing results. So if it says you're at a high risk for cardiovascular disease, health insurances can't discriminate against that information. However, I will say it does not protect you for life insurance. It doesn't protect you for disability insurance. And that's because the courts ruled that medical insurance is a right, but life and disability insurance is a privilege. And I will also say it does not protect those that are on TRICARE. So the military can use that against you in terms of employment, which kind of makes sense if they have that much money invested into you and they're paying for your genetic test results. And now you have an increased risk for cardiovascular disease. They can now use that against you, which, right or wrong, I don't know. I can see it both ways. And then the final one is kind of more of a health disparities problem. But if you guys remember, or if you've talked about the Human Genome Project, which basically claimed that which it kind of did, sequenced our entire genome in 2003. There's definitely, I would say we have like 8 to 10% left to do, because remember I mentioned that GC rich regions are incredibly hard to sequence, and areas that have highly repetitive sequences are also super hard for us to sequence. So I wouldn't say it's like completely done, but it's probably as close as we're going to get at this point anyways. But why is this a problem? The biggest problem with this is that it was pretty much made off of people with European ancestry. And so remember I said genetics is a very ancestral, ethnic driven field. And so if you don't have diversity in your reference sequence, it's not going to be as accurate for people that are not white, basically. So African Americans, Asian, Native Americans, their genetic testing will never be as accurate as someone of European descent, simply because the reference sequence is based off of European ancestry. It's definitely a problem. It's something that the field is working to improve and to diversify the reference sequence, but that's most definitely a limitation of the field right now. All right, so what can you guys do as nurses for your patients? I cannot stress how important informed consent is for a pre-test counseling. Providers, doctors, PAs, all of our medical professionals are great. They're not quite as seasoned in ordering genetic testing, so different labs have different methodologies, and those methodologies determine what conditions can be picked up and what can potentially be missed. They're also incredibly busy people, so they don't always have time or quite realize the implications that genetic testing results can have for patients. And so communicating that risk to patients prior to a test is absolutely crucial. And then, like I said, in cancer, the patient isn't always the first person that needs to be tested. A lot of times in cancer, we need to test an affected relative before we actually want to test the patient. So that way we know we're either getting a negative result or like a true negative result, or did we just not find the mutation? So that's kind of a big piece where informed consent and collaborating with geneticists or genetic counselors can be very, very beneficial for your patients. So as a nurse, from my perspective, what can you guys do to look out for your patients in the future? Help recognize when genetic testing can be useful. So those red flags for breast cancer and colorectal cancer that I talked about, recognize when those family histories are kind of might be benefited by having genetic testing done. It's absolutely not for everyone, but assessing family history and then referring those patients to us to have that first conversation is always the first step. Understand that we are getting better and better every day with our genetic testing, but there are still huge limitations that we have to our testing. I do think that's going to continue to change over the future, but there's still a lot we don't know. And then utilize your genetic counselors. Of course, I had to throw that in there, but really, with how much the field is taking off, are the need for genetic counselors in hospitals and clinics and all these places that genetics is being utilized. I can't stress that enough, simply because it's kind of our job to take some of that responsibility away from the providers to have those deep conversations that need to happen prior to a test. So that way patients are fully understanding what they're consenting to before that test, and then what those results could potentially mean for them.
Awesome. Well, again, thank you very much for taking time out of your day today. I know it was it's very precious to you in your current role. So I thank you for taking the time and speaking with the students today. I know a lot of them have a big question mark over their head and are wondering what to pull out of this. But again, I reemphasize the fact that this is kind of an introduction. They're not going to be genetic experts, but it's just to help them to see if they notice this type of testing results, that it's not just gibberish, that it does make some sense. So
absolutely. Thank you guys for having me. I appreciate it. Hopefully it wasn't too overwhelming.
Thank you.
all right, I'm gonna log off. Thanks again.
All right, sounds good. Thank you, sir.